This video is going to be covering how to record your observations for your Wisconsin Fast Plant Labs. Now, I can't stress to you how important this is because at the end of the unit, you're going to make a big lab write-up, and if you don't have proper observations taken, you're not going to be able to accomplish and complete that assignment. And additionally, I'm going to be checking your observations and grading you on those as well. And I don't want you to be caught off guard. I want you to have nice detailed observations where you can get an easy A from me. And again, be ready for that end assignment where you have the data you need to be able to complete it. So our overall objective is just to understand how to make proper lab observations, during our Wisconsin Fast Plant Lab. And our essential question is simply, what does a Wisconsin Fast Plant observation look like? So you guys have this observation sheet available to you, given to you in class. Now this is just a snippet of the stacked version where you have several of these stacked together. The first thing you need to do always is put down your date, and then we have 4 different observations we're making. We have 2 controls, C1 and C2, and we have 2 experimental groups, E1 and E2. The height is the first thing you do. You want to go in there and you want to make an observation height. Now you have your barbecue bamboo skewers that we measured off, in centimeters, and marked them with a marker, then put them into the planner quads. So really what you should be doing is just looking at the height in reference to that and just estimating based on that. I don't want you grabbing a hold of the plant's stems because they're super fragile and you'll end up snapping a couple and you're going to kill the plant and you're going to mess up your lab. So let's avoid touching the plant at all costs if we can. Then you want to make a detailed observation, something written out where you're saying, hey, this is how many leaves there are, how many buds, how many seed pods. This is the general color of the plant. You want to have this kind of detailed description, especially leaves and buds and flowers and seed pods, and you can even go into a number of petals. But you want this kind of detailedness because you're going to be comparing and contrasting your control group to your experimental group. And these minute differences are going to make the difference in proving whether or not maybe that additional fertilizer was beneficial or if it was harmful and so on. So you really need to have these very detailed observations where you're taking numbers, quantitative numbers, you're measuring things, and then you also have those qualitative observations of light color. Then the next thing you'll do is you're going to make this into 4 squares. You label them C1, C2, E1, E2. You put your drawing there, detailed drawing of what's happened, and then you're going to repeat the process with C2, process again repeated with E1, then lastly repeat the process with E2. This takes a while. This is detailed. You notice that these observations that are written out here, this is not just plant hasn't grown or plant's taller. We're not concerned with that. Really lazy observations. We need something with a lot of detail and meat to it. It's going to pay off in the end when you're doing your final lab write-up, and it's going to pay off when I check your lab notes. And so don't set yourself up for failure. Make sure you're taking the time and doing the assignment correctly. And if you have any questions, I'd be more than happy to help you out in class. Look forward to seeing you, and take care.